There we go. Okay. That webinar was made by a group of people who developed this exhibit. Grady and Addie, I'm going to start. It was developed by people who made this really amazing exhibit at Smithsonian Natural History called Outbreak: Global Epidemics in a Connected World. And if you get the chance to go to the Smithsonian in the next 2 years and go to natural history, you should go to that, because they had all of that set up before Corona even happened. And it's about the stuff that the guy talked about in the webinar, like how do we work together with other countries to prevent pandemics. It's just really great. And last year, my track for DC was I took students to the exhibit, and it's like totally dorking out. It was great. But we don't actually have that many slides about epidemiology, but I want to kind of relate what they talked about in the video with what's going on in the world right now with epidemiology. So epidemiology is studying disease in populations. Studying disease in populations. And by studying it in populations, you can come up with better strategies for prevention. So if you get into nursing, not invention, prevention. If you get into nursing and you like the theory about disease, but you don't really like the hands-on, you might consider epidemiology or public health as an option. We actually have a couple of our bio majors from the past 2 years. They're doing masters in public health at ETSU and loving it. One of them is focusing more on environmental health, and the other one more infectious disease health. So doing epidemiology and studying public health, it really depends on getting data like so, all different kinds of data about where is disease happening and how many people are infected or affected, and determining trends in populations. So there's a lot of statistics involved, and there is a lot of data analysis involved.
So that's where we get like the quarantine days in, right? For how long you have to.
Yeah, so your quarantine dates and how long of a time you have to be quarantined came from data from the early days of coronavirus infections, and looking at, if we go back and look through how this disease spread, can we determine what's the proper quarantine dates? So there are major levels of organizations that do this kind of research, and they're at all different levels. So I'm going to start from the biggest level and kind of go down to local level. Maybe I should try to write this one here. So the major organizations that work with public health are as follows. So we have World Health Organization, or WHO, World Health Organization, and you may have heard about World Health Organization lately, and if you watch the guy's webinar, he is most disappointed with public health during this pandemic at the world level, like that there's no cooperation. And he's right about that. And if you heard that the US government said, we're not giving World Health Organization any more money because they have disappointing, that is true as well. And I would agree that they have been disappointing. I'll come back to that in a minute, but that's the biggest worldwide epidemiology group. And then when you get into the US, what's the US Public Health Epidemiology? CDC. Yeah, so in the US, we have Centers for Disease Control and Prevention. But people still abbreviate it CDC. The prevention part didn't get added on until about the last 15 years. But CDC is at the US level. And you may also have heard that the CDC has messed up its recommendations and they have not been consistent, and CDC has not been perfect during this pandemic either. That they are our national epidemiology group. And then you get down to state health departments. And here in Bristol, we're under what Tennessee prescribes and what Virginia prescribes are different, which is kind of interesting. And then you have local health department. Health, not Heath. Health department. So when you if you worked at Bristol Regional Medical Center these days, do any of you actually? Some years I've got people that are already working there. And if you have patients that come in and they test positive, you have to report that to local health department, and local health reports to state health department. State health department reports to CDC, and I'm pretty sure the CDC is still reporting to World Health Organization as well. So that's for the tracking and the data. So in this system, you have patient level data that has to be reported up the chain. And for certain diseases, it's a requirement that you report up the chain. If people get coronavirus, or if people get hepatitis, or what are the other reportable diseases, like mono, there's this whole list of reportable diseases that have to be sent on to the next level so that epidemiologists can track who is infected in real time. And this whole system depends on cooperation. This whole system requires cooperation if it's going to be effective. So early on, this outbreak started in China. China was not fully cooperative with World Health to let them know exactly what was happening as it was happening. What tools does World Health Organization have to make any given country tell them something? Yeah, they don't. There's not much enforcement power of World Health Organization, because each country that belongs is an independent country, and they cannot make countries do anything. It's kind of a goodwill. Let's work together. Let's share our data. Let's protect the health of the world. But there's not a lot of enforcement there. That said, there is not an alternative to World Health Organization. There's not like the other group that we could turn to. So if the U.S. does not work with World Health Organization, we're kind of on our own. So even though they're not perfect, I feel like we should still work with them, because that's what we've got. And funding from individual countries to World Health can push on them some, but they don't have much power to push on countries individually. It's tricky. Tricky to get this cooperation to happen. I should like update this with the coronavirus heat map. Like, there's a lot in the US, there's a lot in India, there's a lot in Brazil. Europe is starting to go back up. But for people who work in epidemiology, you can have certain specialties. So one of our previous students was a biology major here and security and intelligence minor. And she now works for a company that consults on global biodefense. So they work on watching for like bioterrorism weapons or chemicals related to that popping up around the globe. Cool. So I want to have her come back and speak at King sometime. We have another student that graduated in biology that started working for Department of the Interior, and her job was to monitor how do people get hurt in national parks. That was her job. So she's in charge of slides. Mm-hmm. It's falling off of cliffs is a big one, actually. When we asked her, so what are the ways that people get hurt that you wouldn't think of? Falling off of things is a big one. And like getting bitten by various animals is a big one. But falling was the number one. She's now moved on and she's working for the CDC, but she works in Washington, D.C., and I don't know what she's working on now, but it's not national park stuff. So a lot of cool job opportunities with epidemiology. Some years I have asked people from local health department to come in and talk to us, but I feel sorry for them this year and I'm not asking. My neighbor works for Sullivan County Health Department. She's just really stressed right now. They all are. But this particular page has 4 different definitions that I would like you to know that relate to spread of disease through population. And those are endemic, epidemic, sporadic, and pandemic. So I'm going to probably go in this order. Start with endemic. So to determine how a disease is spreading, you have to know where it's happening and when it's happening. So you need the location and you also need timing to determine which kind of spread is a certain disease showing. You know the location and you know the timing. And in the US, like this is showing, some health department has to report to some overseeing body, like this is how many people have this disease today in the US. So it depends on that reporting. And if you see that there is a certain geographical pattern consistently over time, that's endemic. So endemic is you have a known location where this disease tends to happen, and it's pretty predictable that it always shows up at that rate. So if we looked at this graph and this year, next year, the following year, and it always looked about like this, like people in the American Southwest are getting this disease and it's about the same number of people every year and it's predictable. That's known as endemic. Endemic. Like it's in the same spot, same place. There's about X number of people per year that get it. And endemic diseases depend on an environmental factor. Or let's say, and it's usually depending on an environmental reservoir. So for example, this could represent hantavirus, which is a virus that is spread by rats in the desert. It doesn't happen in places where there's no desert, and it doesn't happen in places where there's no desert rodents. But a certain number of people in the American Southwest every year get hantavirus, and it's just like part of their environment. Around here, like Lyme disease is becoming endemic. It happens around here, and it's kind of predictable how many people will get it, and it's not increasing or decreasing very much. It's just a fact of that environment. That's endemic. This one, sporadic, is something different. So sporadic occurrence, there's no rhyme or reason, and it's totally unpredictable without a pattern. Right? There doesn't seem to be any location associated with the disease, and no pattern can be discerned. It just kind of pops up once in a while. That's sporadic. Think of an example of sporadic. Sporadic, it's usually a disease that's more rare. More rare, and it's once in a while, and it's not centered on a certain location. These don't get a lot of attention. This is not an emergency. This is not usually an emergency. Like, we would like to reduce it, but it's kind of a stable, coming from the environment disease. These two represent more of an emergency. Epidemic and pandemic. So the thing that defines epidemic is that you've got increasing rates at some focused locations. I'm supposed to say locations. Increasing rates at focused locations. And epidemics are defined by accelerating numbers. It's numbers, accelerating numbers. And this particular graphic came from, I think it was syphilis. I think these were syphilis data. In this, what do those places have in common that have the dots? They're big cities. So something about big cities was leading to an increase in syphilis, and this actually overlaid IV drug use. Those two things were related. So as IV drug use increased in big cities, syphilis went along with it because it can be spread blood to blood. I think it was syphilis. So there's focused locations, and it's increasing numbers. It doesn't mean that suddenly millions of people have syphilis. It could be there were a few, and then it tripled in a month or a year. That makes it epidemic.
But it would stay local to that area.
If it stays in the same country, it's epidemic. If it moves globally, then it's pandemic. So we should go ahead and write that in too. Pandemic is an epidemic, it's not going to show up very well. It's an epidemic that spreads across continents. An epidemic that spreads across continents becomes pandemic. The new coronavirus, it only took really about a month for it to move out of Asia into the other continents, because people travel so much now. The goal is always to stop an epidemic before it moves to another continent, but it's difficult. It's very difficult. Oh, what are the other epidemics that people talk about around here besides coronavirus? Seasonal flu. Chlamydia. People don't talk about that around here, but it probably is becoming an epidemic. Most of you are not from here. Only Addie and me. No, Grady, you're kind of from here. Yeah. What other epidemic do we have around here that's just like so increasing? Drugs. So this concept, it doesn't have to be just infectious disease. It can be diabetes, it can be drugs, it can be depression. Like that's probably one that's not, people aren't talking about it as much, but if you looked at data during the pandemic for depression, it would be accelerating as well. So anything that's accelerating, it doesn't have to mean that there's a lot of people that have it, it's that it's accelerating and it's speeding up, then it's epidemic. If it crosses continents, then it's pandemic. Something else I wanted to use this slide to point out is that in that webinar video, they were sometimes talking about a disease called just SARS.
And also MERS.
Yeah, and MERS. They were talking about SARS and they were talking about MERS, which were epidemics of the past. And I want to draw on here, this is just for your information, you don't have to write this down, but where SARS and MERS generally were. So SARS, let me make that a little bit later. Okay. SARS was an epidemic that started in Southeast Asia, and it basically stayed in Southeast Asia. And there may have been like 5 people that made it to the US and 12 people that made it to Europe that had it, but it didn't keep going. So that one was able to be contained. MERS is like Middle East, like that. It's actually not that far into India, but MERS was an epidemic that didn't go so far outside of that region, it didn't become a pandemic. Epidemiologists and doctors were able to stop it before it got so far. That did not happen with our current coronavirus. It got so far, so quietly, that it could not be stopped. So the more deadly a disease is, the easier it is to stop it at an epidemic because people don't feel well enough to keep spreading it all around. Like Ebola. Oh, I'm so bad with African children. Ebola did not make it very far out of Africa because it makes people so sick that they can't travel. And it has mostly been contained to Africa. It didn't spread pandemic. When 98% of, well, let's say when 90% of the people who get this coronavirus feel pretty good, so hard to stop it. So hard. Thus we have the situation we have now, and it will be with us for years. It's not going to go away. It's going to be around for years. But we have to learn how to live with it. If you've heard people say that, we need to learn how to live with it. That's true. It's going to be here for years. We just need a vaccine that's good, and to try to get people vaccinated and make adjustments because the cat is out of the bag or whatever. The virus is out of the plane. I don't know. These four definitions, these will be multiple choice type things on the next exam. That level of knowledge. And all of the epidemiology section is multiple choicey type info. Oh no, I know what I wanted to do with this. Okay, which one of these graphs is most reflective of epidemic? You've got a really steep slope, like a big increase quickly. So that would be most like epidemic. This one, you had a spike and then it went quickly back down. That could be sporadic. This one, that could be sporadic, or that could be endemic. It's almost about the same every year. But to know that something is epidemic, you look at the increasing rate. Or we could say that this started out like an epidemic and then it went back down really quickly. Like that could have been MERS. It was an epidemic, it got under control, it went back down quickly. So it's the rate of spread that let you know whether something is epidemic. And notice here, this is few cases. It doesn't have to be thousands of people getting something to be an epidemic. It just has to be localized and spreading quickly. So we have a little section here about diagnosing infections. And I like to talk about this some because as nurses, you are always at the very front edge of ever diagnosing anything, because it always starts with taking a sample. To really diagnose anything, you always have to take a sample from a patient. And from the semester that you start nursing school, you start learning how to do all these procedures that let you take samples of various types from a patient. This is something I don't know how to do, but what is the procedure for taking a sample for coronavirus testing? Swab? Yeah, now that there's the deep swab and then there's a shallow swab. I do about two. There's some coming that are saliva test. What's the procedure for diagnosing urinary tract infection? Pee in the cup. They can. If they're on a catheter, you have to get it for them. But you glad you're not going to be a nurse?
Yeah, just a little bit.
Yeah. See what else is up there. Sometimes you'll need a blood sample, but there's particular ways that you've got to get the sample, and the technique that you learn is to avoid. You don't want to have cross contamination. But you also have to get enough from the correct location. So all the tricky techniques that you have to learn. You got to start with a sterile instrument and not let it get contaminated, but also get enough of the sample that you don't have to go back and be like, we didn't get enough. I always want an old nurse to do a blood draw on me, or like somebody that's 50. Honestly. The ones in the 20s, I'm like, oh Lord. We have an old nurse.
The older ones are rougher sometimes.
Sometimes they are, but they don't ever have to do it twice, I find.
There was one time my blood stopped pumping in the
Yeah.
tube, and she freaked out. I was like, why are you freaking out, man?
A young one or an older?
Yeah.
Yeah.
And then she walked out while it was still going on. I was like,
Yeah.
Do
Do y'all know the phrase, fake it till you make it?
Yeah.
Like, she should just stay there with you and keep talking.
You're like, so, how we doing today? I'm just
Yeah.
gonna have to stop you right here.
Yeah, if you keep talking, and just talk them through what you're gonna do, and that's good. Did I tell you about Joseph's sample? So when Moses had to get tested, he went in and it was a real medical doctor that was doing it, and she had done a bunch. She was just talk, and she just took that thing up his nose in like half a second and got it. And he was really shocked, but when Joseph went in, his pediatrician, or the one that we saw, he always does training. He's always got a trainee doctor with him, a resident. And she had never done one before. And the doctor was like, let me tell you how you do this. You want to kind of go in level. Just kind of rotate it. Unfortunately, Joseph is young enough that he's just kind of, la la la, he's not listening. But she was probably 5 times slower because she'd never done one before. She did a good job. And she didn't show fear or anything like that. But
Kids can smell that. They can smell food.
at a certain age, not Joseph. He's his own oblivious self. Okay. So once you get the sample, then you're going to incubate, isolate, gather the info until you can identify what that is. And how long it takes to identify what the infection is depends on what kind of tests are available. So this stuff is going to be done in the clinical lab, usually in the clinical lab. Can you think of any test that like you get it right away?
Hydrochloric, whatever. The catalase thing, right?
No, I mean like in the doctor's office. That's true for our lab.
test immediately.
Strep, yeah. So doing a strep test, there are some that you'll do as a nurse where you take it in the back and you do the test. And then you can tell the people right away, you do have strep or you don't have strep. But if the sample has to go to the lab, they're going to do similar types of testing that we do, except they're faster and better at it. So you would first do microscopy and look at what are the shapes, and then you would do gram stain. And this is kind of like a flow chart of what do you do to determine what that strain is. And it's a lot of the same stuff we're doing in the lab. So similarly, when you get your unknown later next month, you first gram stain it and you see, is it gram positive or is it gram negative? And then if it's gram positive, then you would do catalase test, which is like an instant read right there. And then it can start telling you, is it a Staphylococcus strain? Is it Streptococcus strain? And if it was Streptococcus, you would do your further tests there too. If it's gram negative, we have a whole other set of tests that we would do, which we're doing tomorrow. We'll learn those tomorrow. But by doing these and examining the results, you can figure out what is the infectious agent. And if you love lab stuff, but don't so much like the hands-on, These are really good jobs. It's a separate kind of licensure, and you have to go to school maybe like 18 more months or something, but they're really good jobs. If you like doing lab stuff and you don't want to talk to people, they're great. They're great. But if you need to talk to people, don't do this. It's usually down in the basement.
It's kind of cold and freezing.
Huh?
Sounds cold.
It might be cold. But when I was a little girl, I had, well, they still are my mom's cousins. They had these kind of jobs, and they always drove Corvettes. Like, that's what they spend their money on. They always drove Corvettes, and they had worked in the clinical labs for years. And it's good money. And they're kind of medium on the chatty scale. They're good jobs. Pretty much always a need for clinical lab techs. This slide is showing that there are also more advanced methods where you can check DNA tests. You can do DNA tests or polymerase chain reaction to determine what that strain is, like, really fast. We don't do those in our lab, but they're pretty normal in clinical lab practice. But clinical lab techs, they know how to do both of these at once. Did you all know Braden Rassy? He got hired he is doing the labs for coronavirus testing. Starting last week, he got hired to do that. Did he? Yeah. A lot of, I'm learning about, like, so many former students that had it, and they've all gotten better as far as I know. But yep, he's doing it. He's learned to do this, and then when he wants to be a physician assistant, but he needs more patient contact hours to do that. He just let me know last week he's going to do coronavirus lab testing. This slide I'm just going to skip over. I don't feel like doing that right now. I'm going to go to this. Okay. So this is a preview of a test that we're going to do later in the semester that you can do to determine which drugs treat the infection. And this is a little bit like what we did last week in lab when you put that antibiotic disc down on the agar. And then you come back and look, and if you see that a certain antibiotic has inhibited the growth of the bacteria, you call that a zone of inhibition. Zone of inhibition, which means that the drug stops the growth of that microbe. So we actually do this same test later in the semester. And this looks like a picture. I took this picture. Holy cow. This is our crappy lab table. The old scratched up prep table in our lab. We do this later in the semester, but when you do it, you end up coloring the bacteria all over the surface. Kind of like you all last week, you colored the top third and then you put the discs on. For these, you spread the bacteria all over the surface and you put antibiotic discs, and you want to see, like, which ones of these antibiotics do anything or stop this bacteria from growing. So on this plate, that one's having an effect. This one, it's totally pointless against this bacterium. So tomorrow in lab, you get to look at your ones that you did yesterday, and there's certain that work against certain strains. This is called two different names. You can call this Kirby-Bauer test after the people who developed it, or you can call it antibiotic sensitivity test. And a lot of times this this is not done. You gotta wait for the bacteria to grow. So a lot of times, if you think that the patient has a UTI, you would say, I'm gonna prescribe you this antibiotic. Go ahead and start taking it, pick it up from CVS. And then you start this kind of culture and you would check that antibiotic and make sure that it's gonna work. If you see that it doesn't work, then you have to call the patient back and say, stop taking that. Or if it was this one, stop taking that. I'm giving you a new prescription for something that will actually work. This happened to me a few years ago.
That'd be annoying, but big, I just bought
Well,
this stuff.
The alternative is you tell the patient, wait until Wednesday and then we'll prescribe you something.
Yeah.
That's kind of better. But if 80% of the time this would have worked, sometimes you go ahead and get them started on it and say, oh, wait,
Here's a better one.
here's a better one.
Now, so what's the difference between, because the one that you do there from the line around that is real crisp, but some of the other ones, it's kind of like a jagged line. It's
It
not
has to
actual
do
crisp.
with like how big how big is the drug compared to the holes in the agar and how well is it diffused?
Oh, okay.
And this particular bacteria, this is Pseudomonas, and it always grows green like that, which I kind of like. But it will tend to have more hazy borders than some like this. This person didn't color enough. See how there's kind of streaky? This person didn't really color enough. This bacteria, there's a lot of drugs that could treat it. This one, it's only really 2 that are helpful.
So then if you get a drug that's like, it treats it but not as well as another one, would they just usually leave you with that drug or would they call you and have you give the other one?
Usually, you got a Pseudomonas, this is Pseudomonas. Most people that have a Pseudomonas infection, you would prescribe them one of these 2 because those are the usual ones that work. You wouldn't usually prescribe them ones that definitely aren't going to work. But say that you prescribe this, which is the normal, and the plate came back and that was all green, you would go to the next best one. So it's kind of known what are the best drugs for different infections, and there's these huge charts. Or you can look it up on the computer now, but usually you prescribe what's the normal one. Like for ear infections, it's amoxicillin. But if that won't work, you go to the next one, which would be, I don't know. The amoxicillin worked for me, yeah.
So what happens if a person's allergic to that antibiotic and do they just have to deal with it?
You have to ask them on their chart in the beginning, which you all do that as well. Like you go like, here's your clipboard, fill this out. And then hopefully that goes into their permanent electronic record. And then when the prescriber sees that, oh, they're allergic to that, you would not give this, you would go to one that they're not allergic to. If there is another option, which there usually is. Yeah, so all the things that you have to ask patients when you first do their chart, there's always a reason for it. And drug interactions are a big cause of that. Are you allergic to penicillins?
Yeah. Like the whole
Sorry.
family of it.
The whole family. We'll talk about that later when we get to drugs.
Penicillin's the worst, but like amoxicillin, a couple other
Yeah,
stuff.
all of them you wouldn't want to use. Yeah.
What is it, the almond?
They all have a related structure. Okay. So that's the end of that slide set. I am going to go ahead to another one that you don't have yet. Or I'll tell you what I'll do. I'm just going to write on blank screen. Just going to write on blank screen. So let me think of the best way to just get a blank. I know. That way everybody will have the same stuff. Yay. So I want to give some intro to immune system, because that's where we're headed next. Our immune system. And we've got two major branches of our immune system, or two major divisions. And we refer to these as innate immune system versus the acquired immune system division. Both of these are going to help protect us from infectious disease. Both of them are going to help protect us from infectious disease. But the innate are kind of the first set of barriers or processes that pathogens would overcome, or pathogens would face if they're trying to infect us. The innate immune system is the first that pathogens would have to overcome if they're going to infect us. The acquired immune system, we're going to use that if the innate, that says innate, didn't stop the pathogen. So we refer to the innate immune system components as the first and second lines of defense. Your first line of defense is barriers. The second line of defense is processes. And then if the bacterium or the virus or the fungus or the worm gets past those, then our acquired immune system will kick in, and it's our third line of defense. This says third line of defense. Okay, so our innate immune system includes the first line of defense. Try that again. That's weird. Okay, maybe it has to be black. Our innate immune system includes first line of defense, which is what we call mechanical barriers. Mechanical barriers and flushing mechanisms. So on Wednesday, that's the first section of the lecture is first line of defense. Those actually don't take that long to describe. And then we will start on describing the second line of defense, which includes certain processes. So there are chemical barriers, fever, inflammation, phagocytosis, cells eating cells, interferons. I need to spell that one more carefully. You can't read that. Interferons. And then another category that we'll just call antimicrobial proteins. All of that stuff is second line of defense. Chemicals and processes. This is like all going to be, it's all the same, but it's more in detail. When you come to AMP2, you get 2 days on the whole immune system, which is pitiful, but that's how it is. Or you already had 2 days on the whole immune system before. Let me also put up what are the major characteristics of innate immune system. There are certain ways that it acts and certain qualities that it has.
I'm not going to do a third defense.
Third line of defense, I'm not getting to today. It's too far away. We will, but that's on exam 4. That's a ways away. So how would we describe characteristics of innate immunity? Characteristics of innate immunity. You're born with it, and it is always ready. Or it is preset. You don't have to wait around for it to activate. It's already there. And the born with it part, that's where the word innate comes from. Like it is a part of you, it's part of your being. You've got it ready. And because you're born with it and it's already ready, it's fast to act, but it's also weak. It's kind of a weak protection, but sometimes that's enough. So people who, you probably know people, like you heard, so-and-so is sick. They're around these 5 people. Later on, when so-and-so tests positive, and those 5 people, like only one of them tested positive and the others did not, even though it seemed like they were doing all the same stuff. Some of those people, their innate immune system was enough to fight off the virus, and it never progressed to make them sick. But maybe that one person, their innate immune system did not fight off the virus, and they did get sick. So it's weak and it's fast, but sometimes it's enough that it fights off whatever pathogen. Your innate immune system does not require previous exposure. It does not require previous exposure to that infectious pathogen. It doesn't matter whether you never saw it before. Your innate immune system can still help protect you. And it's fast, but it's kind of wimpy. And the final thing to know, your innate immune system does not give immunological memory. This is not the part of your immune system that lets your body remember, oh yeah, I'm supposed to fight that off. That's the acquired part. So you can think of innate immunity as like border. You have to wait and call them. They're kind of wimpy. Could be enough. It's just the first thing that the pathogens need to get past. That's innate immunity. So bring this with you on Wednesday. Be looking for actual slides and quiz for Wednesday. This probably answers a lot of it. this probably answers a lot of your quiz stuff already for Wednesday. And lab tomorrow is another fun one. We're doing gram-negative pathogens.
Cool.
Gram-negative, so they stink, just so you know. Smell bad.
The same way quiz.
I don't think this counts for her because she has a concussion, but
Not sure.
I'm not sure.
That's probably true.